I'm Joseph Mertz, a postdoctoral research fellow at the Wilmer Eye Institute at the Johns Hopkins University School of Medicine in Baltimore, Maryland. The goal of my research is to find genes and proteins to target for neuroprotective-based treatment of glaucoma and other optic nerve diseases. The term neuroprotective treatments is really as simple as it sounds. It describes treatments like pharmaceuticals, for example, that protect neurons from dying and from losing their function. Of course, it gets much more complex with more detail, but that's one reason it's an area of intense research. Another reason we research neuroprotection is because neurons perform innumerable critical functions throughout our bodies, and because many of them cannot regrow or be replaced once they're lost. So protecting them is of vital importance in many diseases. Glaucoma, the world's leading cause of irreversible blindness, is one of these diseases. Over 3 million people in the United States and over 70 million people worldwide have glaucoma in some form, and the chances are high that you know someone with this disease. Glaucoma and the blindness it causes affect people's safety, mobility, quality of life, their ability to perform numerous everyday necessities, and their psychological and emotional health. The costs of glaucoma to the American public, which include direct medical costs, loss of productivity for glaucoma patients, and costs to families and caregivers, are approaching $3 billion per year. Sadly, the number of Americans with glaucoma and its costs to society are projected to rise as our population ages. The blindness and loss of vision that glaucoma patients suffer is due to the loss of a particular type of neuron called the retinal ganglion cell. These cells reside at the back of the eye in the retina, and each one has a long extension that goes all the way from the eye to the brain, and all those extensions make up the optic nerve. It's the retinal ganglion cell's job to collect visual information captured by other cells in the retina, like rods and cones, and transfer that information through those long extensions to the brain. You may be familiar with the fact that high pressure in the eye can cause glaucoma and is often associated with it. The most widely accepted theory, we're still not absolutely sure, is that this high pressure puts strain on the retinal ganglion cells at the point where the extensions exit the eye. This squeezes or stretches them and eventually damages and kills them. And because retinal ganglion cells, as is the case for many neurons, cannot regrow or be replaced naturally, as each one dies, the visual information they carried is permanently lost. All current treatments for glaucoma, whether pharmaceutical or surgical, act by reducing this high eye pressure, but this does not always stop vision loss, and many people suffer from glaucoma with no abnormal eye pressure. So if we can directly protect these neurons, we might be able to supplement or replace pressure-related therapies and save vision for millions of people. To do that, we need two important pieces of information. First, when retinal ganglion cells are damaged or under stress, what signals at the molecular level tip them towards death rather than simply being damaged? Second, how can we stop those signals, likely through a pharmaceutical blockade of this specific process? My colleagues and I use a combination of state-of-the-art technologies to try to answer these questions and other related questions. First, we grow human stem cells and use a process developed in our lab to guide them into becoming retinal ganglion cells. As is the case for much of biological research, there are many aspects of retinal ganglion cells that we can't study in a person, so we recreate them in the lab to study them. Second, because they're in a culture dish, we can carefully and precisely control their genes by CRISPR-Cas9 gene editing, treat them with molecules and conditions that recreate glaucoma, and safely test new pharmaceuticals on them to find new potential treatments. Lastly, through highly sensitive and comprehensive omics methods, we can examine the retinal ganglion cell system as a whole. Omics methods like genomics and proteomics that have been developed in the last several decades have given us the power to analyze all the genes or all the proteins in a cell or tissue at once. Since there are over 10,000 genes producing proteins in a single cell at one time, omics methods are essential to make sure we're not missing important actors in disease processes like retinal ganglion cell death and glaucoma. By combining these approaches, we have identified a handful of genes, many of them unexpected, that play roles in retinal ganglion cell death. If we can find ways to stop those that help cause it or boost those that prevent it, we might be able to find or add a future weapon to the arsenal to fight glaucoma.